right, welcome honors biology students. I'm glad to have the opportunity to talk to you about chapter 9 from your textbook. I know you've seen some videos that I've done before on YouTube, and now I'll be filming some just for you, and that will hopefully help you as you try to understand the material from the chapter that we hope to cover this week. So chapter 9 is the cell cycle. We're at an interesting place in the year where we've finished most of the topics in molecular biology, and we're about to move into talking about genetics. And the pathway between molecular biology and genetics is via the cell cycle. So we need to try to understand what actually happens in a cell when it turns into two cells. So how does it replicate all of its DNA, and how does that process look? And we call this process the cell cycle. So here are some beautiful pictures of cells undergoing cellular division, and I love when we get into these chapters because we start seeing more of these immunofluorescent images. In this case, you can see microtubules are going to be colored in bright yellow, DNA is colored in blue, and you see that a lot of stuff is happening. We'll talk about what's happening in these various pictures, but essentially the blue stuff is getting split in half and divided into multiple locations. So it's really important for cellular division that cells can make more of themselves. We know from this definition of the cell that all cells come from other cells, and we call this cellular division. It's funny that cells multiply by dividing, but we would say that the cells can multiply, cells can divide. It does the same thing. We end up with more cells just like the cells that we had before. This cell division is going to be talked about as part of a cell cycle. So the cell cycle is the process through which a cell goes through growth and development before ultimately dividing into two cells and then repeating the entire process again. It's called a cycle because it happens over and over and over again. So why do cells go through the cell cycle? There are some pretty important reasons. One of those is reproduction. So single cellular organisms reproduce asexually, which means that they just split. That's how they have offspring or progeny. In organisms like humans, growth and development is a pretty important reason that we need to do cellular division and go through the cell cycle. We've talked about this in class before, that all of you start as one cell, and now you're made up of many, many cells. All of those cells have the same DNA in them because they were all outcomes of cellular division of that one cell you started with. And then sort of on a more short-term basis, mitosis, cellular division is seen in tissue renewal. So a great thing to think about is when you cut yourself, the fact that skin can scab over and make new skin in the area that you cut yourself are just the cells on the edges of that cut going through mitosis and renewing the tissue that you have in that area. So we'll jump right in. Chapter 9 is actually, I think, fairly short and straightforward. It has three major concepts. The first of which is that most cellular division results in genetically identical daughter cells. And we've sort of already established this, that if you start as one single cell and you end up being a lot of cells as you're almost an adult now, and all those cells have identical DNA, then it's really important to have genetically identical cells come out of this process. So we have to copy all of the DNA and distribute it to the two daughter cells. And we know by thinking about biology, thinking about evolution, that DNA is passed from one generation to the next generation with remarkable fidelity. It's really well conserved. Not a lot of changes happen as we go from cell to cell. So there's a lot of vocabulary as we start talking about cellular division. And some of this we've already used, but I'll just run through it so that we can make sure that everybody's comfortable. When we talk about the genome, a cell's genome is all of the DNA in its cells. So the Human Genome Project aims to define all of the genes that are inside of humans. The genome is a generic term that we sort of talk to about the idea of all the DNA. And then when we talk about the physical structures themselves, we will often talk about chromosomes. Chromosomes are DNA molecules inside of the cell. And that's when the DNA is packaged up really tightly so that it can move around. So humans have 46 chromosomes. We have 23 pairs of chromosomes, and that's where you hear the 23andMe when we talk about sequencing someone's genome. The chromosomes are composed of chromatin. So chromatin is a sort of loosely packed DNA and protein complex. We've talked about this a few chapters ago. And that chromatin can get packed up more tightly into chromosomes in various parts of cellular division. You'll notice a lot of things are called chroma this and chroma that, and we'll talk about in a little bit that's basically vestiges of how this original science was done. Basically, things were dyed, and the dyed things were called chroma whatever, and the not dyed stuff was called something else. And then over time, we've had better ability to understand what those things are, but those names stick. And I apologize, it is a little confusing when we talk about chromosomes and chromatin. In a minute, we'll talk about chromatids. The same kind of prefix keeps showing up. So every eukaryotic species has a characteristic number of chromosomes in each of its nuclei. In humans, that would be 46. I'm going to pretend that I can rattle them off the top of my head for other organisms, but you guys can look that up. Other organisms would not be 46. Some are going to be. It's sort of a characteristic number that's different for every organism. It actually doesn't align necessarily with how complex or how simple an organism is. There's a lot of still mystery around why species have their own number of chromosomes, although the answer is ultimately going to be evolution there. There are two types of cells when we talk about cellular reproduction, somatic cells and gametic cells, or gametes. So somatic cells are those cells in your body that are not used for reproduction. They will all have two sets of chromosomes. So those are the copies of chromosomes from your parents, your mom and your dad. And then you will create some gametes, which are reproductive cells. So if you're a male, you create sperm. If you're female, you create eggs. Those have one set of chromosomes. That creation of a cell with one set of chromosomes is going to be the next chapter when we talk about meiosis. And that's actually critically important for sexual reproduction, because when you want to combine a sperm and an egg, you want to get back to the set number of chromosomes that you expect to have. So you actually have that DNA from the outset. Forget about gametes for right now. We're going to really, in this chapter, stick with somatic cells, the non-reproductive cells, and how they reproduce. So here is a beautiful picture of a cell. This is not an immunofluorescence. This is just with a dye, and you can see the darkest part of the picture. These blue squiggles are going to be the chromosomes, and they're named because when they were dyed, they were threads that appeared to be dyed, chromosomes. So when we want to divide a cell, you've got to think that if we want to put the same stuff in every cell, we have to copy all of that DNA. So in preparation for cell division, DNA is replicated, and the chromosomes become these chromosomes as opposed to chromatin. Chromatin would just look like a blue filling in here. Each of these duplicated chromosomes, so after the DNA is replicated, we call it a duplicated chromosome. It has two sister chromatids. So two chromatids make up one chromosome, and they're joined at a structure called the centromere. So centromere is the structure that joins two chromatids together. And when we want to look at a picture of this, here are two sister chromatids. You can always see there's like a little bit of an indent where the centromere is. This is actually a protein complex that's holding these two sister chromatids together, these two DNA molecules together. And that's where we will often talk about a structure of a chromosome looking like an X, although it's actually more like an H. This goes with this, and there's a connector between them. So we can very simply think about the process that has to occur for a cell to divide. We have to take a chromosome. We have to duplicate that chromosome. That duplicated chromosome is now made of two sister chromatids. And then we go through a process where we take one sister chromatid and go this way, and one sister chromatid and go this way. To make things really confusing, these chromatids, when they're by themselves, are now chromosomes again. And then this will repeat, because that chromosome will duplicate, become a duplicated chromosome, and then it will move into two more cells. So sister chromatids always separate, and they go into what we call daughter cells. And if you're wondering what one daughter cell said to the other daughter cell when they stepped on her foot, she said mitosis. So eukaryotic cell division consists of two processes. See, I'm really proud of that joke there. Mitosis, which is how we divide the genetic material, and then cytokinesis, the division of the cytoplasm. So as we start thinking about this, I want you to think about two processes. Mitosis is really the division of the nucleus, and cytokinesis is the division of everything else. So you can divide the nucleus without dividing the cell, and that cell would end up having two nuclei inside of one cytoplasm. You can think about why or why not that would be a good idea. But we're going to talk about mitosis, and then we're going to recognize that cytokinesis also occurs, dividing everything up. All of your endoplasmic reticulum, your lysosomes, your Golgi, all of that. Gametes, like I said, chapter 10, totally different process. It's a variation of cell division called meiosis. Those cells in meiosis are not identical, and they have half of the DNA, and we'll come back to that next week. So right now we're really focused on mitosis and cellular division that's going to create identical cells. So 9.2, the mitotic phase alternates with interphase in the cell cycle. So this is where it gets a little bit interesting, thinking about the process of science and the development of microscopy and tools to understand this. So mitosis was identified in the 1880s. And as I already mentioned, an anatomist, Walter Fleming, made dyes that he could observe chromosomes, naming them chromosomes because he could see them. And so he basically had two observation points within a cell. He had times where he saw chromosomes, and he had times where he did not see chromosomes, and instead he saw chromatin. And so in thinking of that, he kind of divided it into two processes, or scientists of the era divided it into two processes. You had mitosis, the division of the nucleus, and then you had interphase, which was everything else. So you had this important phase that you were looking at called mitotic phase. Stuff is really happening in the cell. It looks really interesting. And then to their mind, interphase was everything else. It wasn't really interesting. It was in between the mitotic phases that were worth looking at. So the interphase was just a period where they said, well, nothing is happening. As you've heard in other narratives this year, a lot of stuff is actually happening during this time that early scientists thought nothing was happening. Interphase is important for cell growth. It's important for replicating the DNA. And it's important for doing a lot of preparation to make sure that the cell is ready to divide. But if we look at this simple diagram, interphase is most of the time that a cell is sitting around doing stuff, and then mitotic phase is a relatively short period where it divides. Roughly in human cells, the cell cycle, which would be this entire circle, is about 24 hours, and the mitotic phase takes place in about an hour. So much shorter time for mitotic phase than for everything else that's occurring. Interphase, which is in different cells about 90% of the cell cycle, has 3 subphases, and these are also named pretty generically. There's when they looked at interphase, they saw that there was a point in time where DNA was being replicated, and they thought, okay, the DNA replication is pretty important, so let's call that S phase for synthesis of DNA. And then you know what? We have a period of time before synthesis. That's a gap. Let's call that gap 1. And then we have a period of time after synthesis that's called gap 2. So we do mitosis, we have a gap. We synthesize the DNA, we have a gap. Then we do mitosis again. And in a continuing story that I've already kind of said, the G1 and G2 later is identified to be pretty important as well. G1 is important for growing, replicating, duplicating all of the organelles in a cell, having a cell grow to the right size so they can divide. And G2 is pretty important for doing some final checks, making sure that all the DNA is proofread, done appropriately, ready to go. The cell is growing the whole time so that it can be prepared for mitosis. So G1, S for DNA synthesis, and then G2, and then you go into mitosis, followed very rapidly by cytokinesis. So we can further divide this when we study mitosis properly. So when we look specifically at mitosis, and if we start studying the beautiful immunofluorescent images that we can get of the DNA, what it's actually doing during mitosis, it's going to go through 5 stages. But I want to caution you right now that we're going to divide these into 5 phases that you guys can think about and identify, but know that this is a continuous process. So these are really arbitrary designations that scientists are using to identify sort of what's going on. But prophase goes right into prometaphase, right into metaphase, right into anaphase, right into telophase, such that you can have prophase, prometaphase, prometaphase, phase, proanaphase, meta-anaphase, anatelophase. There's all sorts of combinations in between these where you're kind of halfway in between one and the other. And then as you get towards the end of mitosis, cytokinesis, rather than sitting separately like it's showing you in this diagram, actually overlaps. And so they're both kind of happening at the very last minute. So let's see if we can get this video to play. We might not be able to. Here we go. So here you can see a video of a cell. This is a light microscopy, but you can see those chromosomes clearly through some optical tricks. They're forming up. You can kind of see that they're in pairs here, and they're forming along a central line so that they can organize themselves to be ready for division. And once they all make it into the center, you will see this cell will very quickly pull everything apart, and now we have one set over here and one set over here of chromosomes. Those chromosomes will decondense back into chromatin, and now we have 2 cells, and you saw that they did cytokinesis very rapidly as that finished up. So that's the general process here, and I think you can get bogged down in all the individual steps, but if you think about the processes and how they kind of just flow together, it's going to help you understand things. All right, so let's look at the various processes here. So we will start always by thinking about interphase, and as I said, in interphase, your DNA is decondensed. It's in the form of chromatin. It's just spread out all over. And as we move into mitosis and into prophase, one of the first things you see is that the DNA will condense into chromosomes. So you can see chromosomes forming in prophase, and also during prophase you'll start seeing the mitotic spindle. The mitotic spindle is composed of microtubules. These microtubules come from the centrosomes, or the microtubule organizing centers, and they spread out to really help pull the DNA apart. So you can think about DNA as a thing that needs to separate. Mitotic spindles are the things that grab it and pull it apart. As we go from prophase into prometaphase, you'll see that the nuclear envelope actually completely falls apart. So we have no nuclear envelope anymore. The mitotic spindles will start attaching to the chromosomes at a space called the kinetochore. And the goal here, these chromosomes are going to be moving back and forth and back and forth and back and forth, and they're trying to attach to a microtubule from both centrosomes. So it wants to be attached to both so that you can move from prometaphase into metaphase, where the chromosomes will all align in the middle at what we call the metaphase plate. Now, how do these align? They align because if you think about it, if I attach to one MTOC and I attach to the other MTOC, and both of these are pulling with an equivalent force, we're always going to end up in the very middle of the cell in the metaphase plate. This is an important checkpoint that we're going to talk about in the next section, because when you get to this checkpoint, when all of these guys align, chemical signals occur in the cell to have these sister chromatids release each other, and that force that's being pulled on by the microtubules, as soon as the sister chromatids are released, will simply pull them apart in a process we call anaphase. And then as they pull apart, they will be pulled to opposite poles of the cell. The cleavage furrow of cytokinesis will immediately start. The nucleus will reform, and you'll end up with a set of two cells that are genetically identical. So we can look a little more closely at the mitotic spindle. Like I told you, it's made of microtubules. It comes from the centrosome, which is a microtubule organizing center, an MTOC. And what's cool about this is that the microtubules actually attach to the chromosomes at a structure called a kinetochore. And there was a lot of work done to try to understand how does the microtubule actually pull the chromosome back towards the pole. And when scientists looked, they tested these microtubules, and they watched cells undergoing the process, and they would mark certain parts of the cell. And what they found was that when you mark a part of the cell and then you watch these chromosomes come back, that mark doesn't move. So that might not seem like much, but if that mark were to move, especially if it were to move towards the pole, it would suggest that the microtubules are being pulled in like a piece of rope. Because of this result, what it actually showed was that the microtubule, as it attaches to the chromosome at the kinetochore, actually falls apart, and the kinetochore surfs backwards on a falling apart microtubule. A really cool visual to think about, that this is what's causing all of your cells to divide with pretty high fidelity, not something being pulled, but the chromosomes themselves kind of surfing down a microtubule as it falls apart. To think more about cytokinesis, so this is going to occur towards telophase as things fall apart, as the chromosomes come apart. We will call it in animal cells a cleavage furrow, and that was where the cell sort of pinched in to make two cells. If you think about it, plant cells are going to have to be a little more complicated because they have a cell wall. So they actually will form a cell wall in between called a cell plate. And we can see this here in an animal cell. Really easy in an animal cell, a cleavage furrow just pinches the cell together. So you contract, and then you have two daughter cells. A little more complex in a plant cell because as you want to divide, you have to bring in vesicles with cell wall components. That cell wall will form, calling the cell plate, and then fuse with the sides, making two daughter cells. If we go really simple, simpler than any of the eukaryotes, you can think about bacteria. And in bacteria, we call the process that's analogous to mitosis binary fission. And binary fission, while it sounds really complicated, it's actually even easier than mitosis. The DNA replicates, as you might expect. That DNA attaches the two ends of the cell, and then as the cell splits in two, it just pulls that DNA apart into two cells. So while this is really simple, it's interesting to think about from an evolutionary standpoint that this would be the ancestor to the complex mitosis that we see now, still trying to accomplish the same task of duplicating the DNA and then spreading it across into two directions. All right, so the last part of this process that we want to talk about is how do we regulate the cell cycle in eukaryotes. So as we've talked about with a lot of things, right, we have a process that's relatively straightforward, but the key to that process is when it is activated, when it is not activated. We clearly don't want cells to divide all the time. Why is it, for example, that when you cut yourself, those cells only divide enough to fill the cut? They don't keep growing beyond that. And we know that these regulation occurs at the molecular level. It's interesting here to consider cancer is a disease that's caused by cells that have escaped the controls of the cell cycle. So at the very simplest definition, cancer is just uncontrolled cellular growth that's occurring by cells that are no longer following the rules and the regulation that the cell cycle is trying to impose. So we know that there are chemical signals that do this from some pretty interesting but somewhat simple experiments in terms of understanding all of the processes. And this experiment shows that if they take a cell that is in S phase and they take a cell that is in G phase, G1, and they fuse them together in a laboratory, both of the nuclei that are in this new fused cell will both be in S phase. So we have an S and a G, we put them together, everybody's S. Similarly, if we take an M and a G1, we fuse them together, everyone is then in M. They're both doing mitosis. So the conclusion that you would take from this is that there is some molecule inside of the S cell that says we should be synthesizing right now, and it tells this cell to synthesize whether or not it's ready to synthesize. There is some molecule in the M cell that when it's spread into this nucleus that was in G1, that nucleus goes into M even if it's not ready, because that molecule says it's time to go into M phase. So we really think a lot about these molecules and how these molecules regulate the cell cycle. So there is a cell cycle control system, and there'll be a diagram on the next slide that shows this is like the dial on a washing machine. So there are specific checkpoints, and I'll go ahead and show you that picture. There are specific checkpoints as it goes around this dial. You saw this picture before, G1, S, G2, M. As it goes around, these checkpoints will stop until the cell is ready to go. So if the cell isn't prepared to go from G2 into M, this checkpoint is going to stop it and make it wait there. If it's not prepared to finish mitosis, it's going to stop here. If it's not ready to go into synthesis, it's going to stop here. So these checkpoints are really important because if something's not right with the cell, it will stop. If it doesn't go right for a long time, that cell will actually go through a process called apoptosis or programmed cell death, so that that cell doesn't replicate inappropriately. The first checkpoint that we would see as we go through the cell cycle would be the G1 checkpoint. This is the checkpoint that makes sure that the cell is big enough and that the cell is ready to do DNA replication. This is actually a checkpoint that a lot of your cells hit and stay at, and that's totally fine. So if they don't have the go-ahead signal, if the signal's not there to divide, the cell will stay here and actually enter a process called G0. So if you think about cells that are in your body that have been there for a long time, especially your neurons, for example, they are all existing right now in G0. Some cells can exit G0 and go back into the cell cycle. Your neurons would not be an example of that. As you know, if you damage nerve cells, they cannot regrow. You're not able to just kind of replicate again. So this is called the G1 checkpoint. It checks whether or not the cell has grown large enough for it to be able to divide. The G2 checkpoint, I don't have a slide for, but the G2 checkpoint is important for making sure that that replicated DNA has been replicated with high fidelity. So think about all the proofreading processes that we talked about when we talked about DNA replication. Those take some time to process. And so there are signals that let the cell know that if there are still mistakes in the DNA, if there are still errors that need to be proofread and fixed, to not allow it to go into mitosis, because once it goes into mitosis, all of those processes will have to stop. The next checkpoint is the M checkpoint, and this one is really key because this is the one that makes sure that all of the chromosomes are aligned at the mitotic plate before the cell starts to divide. So you can imagine that if all of them are at the mitotic plate except for one set, and the cell would divide anyways, then the daughter cells are not going to have an equal amount of DNA. One of them is going to have an extra chromosome, and one of them is going to be missing a chromosome. We'll talk about this some more in meiosis as well, but anytime the M checkpoint isn't appropriately managed, you can have pretty significant damages to the cell. So there are also, along with these internal signals that I just talked about that do checkpoints, there are some external signals, and we'll go through this pretty quickly just so that you have some familiarity with this. One of those external signals are known as growth factors. So these are proteins released from some cell in the body to go to another cell in the body to tell it to replicate. One example of this is platelet-derived growth factor, which is an external signal that will tell cells to divide. So a lot of cells in your body are waiting for signals to come from somewhere else before they divide. We can see this in an experiment if they take some connective tissue, cut it up into small pieces, and put it in a test tube. If you add PDGF, a lot of them will grow. If you don't add PDGF, they will just sit there. They won't grow. These guys have not received an external signal to go into the cell cycle, while these guys have. Some other external signals that become important when we think about cancer are density-dependent inhibition. So many cells will stop growing if they're surrounded by other cells. So if the density is high enough, they will stop dividing. And anchorage dependence, which is where cells have to be attached to a substratum to divide. So two of the hallmarks of cancer cells is that they stop paying attention to density-dependent inhibition. They will grow even if there's no space for them to grow. That's how you get tumors. And they also don't have anchorage dependence, so they will grow anywhere. They won't limit themselves into the area where they are already found. So you can see anchorage dependence, they have to be growing on the surface. Density-dependent inhibition, they will form a nice flat layer and no more. If you cut a little hole in there, they will fill in that hole and then they'll stop growing. They will only grow to a certain density. Whereas cancer cells will grow all on top of each other. They don't have to be attached to the bottom surface. They will fill this entire dish up, even starving themselves of food, and ultimately killing the entire population. There's no coordination between growth and resources. So, just to touch a little more, I kind of said this, cancer cells don't respond to signals that would regulate the cell cycle. They don't need growth factors. They make their own growth factors. They do whatever they want. They have abnormal cell cycle control systems. A lot of these come because of mutations in the proteins that would normally carry out the checkpoints, which is a really interesting set of circumstances to study and try to figure out. Normal cells becoming cancerous cells is called transformation. So when we talk about the mutations that occur in a normal cell to make it cancerous, we would say that cell has become transformed. And just to give you guys some vocabulary that you've probably heard before, benign and malignant refer to sort of not threatening, abnormal growth, but that's not moving throughout the body as a benign tumor. Malignant tumors, malignant meaning bad, will invade tissues and often undergo metastasis, which is where they move throughout the body and form additional tumors. So benign tumors are usually no big deal. They can be removed. Malignant tumors usually require more significant medical interventions, especially if they advance to metastasis. So that was everything of Chapter 9. We talked about three major things. One is that cells have to divide and make identical copies of each other. Two is that the process of mitosis will move us through steps to take chromosomes and split them evenly between cells. Along with mitosis, we have the entire cell cycle that includes interphase, cellular growth, DNA synthesis, and more cellular growth. And then the final was talking about checkpoints, the G1 checkpoint, the G2 checkpoint, and the M checkpoint that will help keep this process moving. Hopefully this makes sense. We will have time during our class meetings to go over any questions that you might have. So read the chapter, watch this again, and hopefully I look forward to talking with you about mitosis soon.